In today's video, we're going to be talking about the new COVID-19 vaccines, the Oxford AstraZeneca vaccine and the Pfizer BioNTech vaccine. We'll be talking through what they are, how they work, and answering some common questions along the way. So how do vaccines work in general? Well, vaccines work by triggering an immune response within the human body. And they do this by introducing something that mimics, say, for instance, a virus into the body. That then triggers an immune response. Antibodies are developed. These antibodies then remain in the immune's memory and then are brought out to fight an infection if the body is ever exposed to it in the future. And typically, there's 3 or 4 ways this might be done. One is by using a weakened version of a virus. The other is by actually isolating a particular protein on a virus and introducing that protein as part of the vaccine. And then the more novel, newer ways of using genetics, DNA and mRNA. So the Oxford and the Pfizer vaccines work in a similar way in that they provide instructions through this vaccine to the human cells to develop something called the spike protein. And this is the spike protein comes from the name or the appearance of the coronavirus itself, where you've probably seen lots of pictures of these little bobbly coronavirus with these little spiky bits sticking out, and that's what we're referring to as a spike protein. And by recreating this spike protein in the human body using these genetic instructions, an immune response is created, antibodies are created against these spike proteins, and thereby their future immunity is developed. Now, the two vaccines differ in terms of how they deliver those instructions. So the Oxford AstraZeneca vaccine delivers the actual some DNA into a cell's nucleus that contains the instructions to make that spike protein. The DNA then unravels and then becomes something called mRNA, and it's the mRNA that then has the actual instructions then to create this spike protein. The spike protein is then created by the cell, and then once that spike protein becomes apparent, the human body then sees that as something new and different, and antibodies are created against it. The DNA is delivered into the cell via another virus, an inactivated virus called an adenovirus, and this virus causes the common cold in chimpanzees. However, what's been done is that the virus has been modified to stop it replicating inside the human body, so it cannot actually infect anybody. So the only use of that virus is to deliver the DNA into the cells. The Pfizer vaccine works differently, so it eliminates the DNA step. So it goes straight for the mRNA. So what's happening is you've got this lipid bubble. The mRNA has been created synthetically in a laboratory. It's inserted into this lipid bubble, and then once that's injected into a person, that lipid bubble then goes and attaches onto a cell. The mRNA enters the cell, doesn't go into the nucleus, and that mRNA then has the instructions to create these spike proteins and thereby trigger that immune response and antibody production. So how are they developed so quickly? So normally vaccines can take many years to develop. These vaccines, including the Moderna vaccine, have been developed over the space of around 12 months. There's a few reasons for this. One is that these vaccines don't rely on needing to take a virus and weaken it or identify the particular protein and then trying to recreate that protein in a laboratory and then produce lots of those proteins and as part of this vaccine. These Oxford and Moderna and the Pfizer vaccines, because they're genetic type vaccines, they're really easy to develop. So within the space of a few weeks of the coronavirus being identified, the one that causes COVID-19, scientists around the world were able to produce the genetic sequencing of the virus and thereby able to develop the genetic sequence of the particular spike protein. In addition, prior to COVID-19, we've had similar coronaviruses in the past. So SARS is a coronavirus. There's a lot of work and a lot of research done in the past on developing vaccines for SARS. And also there's been work done in the past in terms of, should there be a pandemic in the future due to this type of virus, what kind of vaccine might we need? So all of that research, those clinical, that understanding was there previously, which sped up a lot of that initial process. And then the fact that we've been in a global pandemic lockdown situation, there's been a huge global effort to develop a vaccine, so there's a lot of resources being thrown at this. Lots of volunteers have come forward to go through the phase 1, 2 and 3 trials. The regulators have worked closely with the vaccine developers to review findings and go through the processes of approving as the vaccine's being developed. And then finally, obviously, the governments, again, because the vaccine was being tested, but actually we knew that this vaccine, this is the one that actually would work, and obviously if the test showed that it didn't work, they would have done something different, but it was, the chances are the vaccine that was being tested was going to work. So lots of supplies were ordered in advance. So rather than waiting for each step to complete before going on to the next step, multiple steps were done at the same time, thereby reducing down the time taken overall from concept to delivery of these vaccines. Now, what are the side effects of these vaccines? So it's important to remember all medication, all vaccines will have side effects, and both of these Oxford and Pfizer vaccines have similar side effects. So very commonly they can cause pain and irritation at the injection site. They can make you feel a bit tired, with some headaches, muscle pains, etc. These things tend to last just a few days and will settle down, and you can take some paracetamol for them. There are some more uncommon and rare side effects, such as changes in appetite, enlargement of lymph nodes, especially to near the site of injection, and some rare cases, especially with the Pfizer vaccine, drooping of the face, and also there's been these allergic reactions as well. So it's important to remember that side effects are possible. However, there's the common side effects, the rare ones are rare, and they do tend to settle down. In terms of allergies, it's important to make sure that if you do have any history of allergies, that you check the ingredients and you double check those with your clinician. So what do they contain? So again, looking at the ingredients or the excipients, there's nothing really different to normal vaccines. So the lists on both the Oxford and the Pfizer vaccine, at the bottom end of the list you've got the potassium chloride, sodium chloride, etc. These are all stabilizers, so they're stabilizing the acidity, the pH of the vaccine. The sucrose, again in both vaccines, that's there as a stabilizer as well. The difference is that the Pfizer vaccine has these lipid molecules, and these are essentially fat molecules. What they do is they create this protective bubble, protective ring around the mRNA to stop it from being damaged and destroyed either in transit or during injection by the blood, etc. But once the mRNA enters the host cell, these lipids dissolve and are excreted by the body. You must have heard that the Pfizer vaccine, because of this mRNA, is quite unstable. Therefore, it has to be kept frozen at -70 degrees, and then it's thawed and can only be then kept at the thawed temperature for a maximum of 5 days. The Oxford vaccine is far more stable and is stored at a normal fridge temperature of 2 to 8 degrees. Both vaccines are given intramuscularly, and both vaccines require a second dose to be given. Now, moving on to our common questions. So, are DNA or mRNA vaccines new? Well, yes, they are. This is the first time that these types of vaccines have been approved for use in humans, including now the Moderna vaccine. However, the technology is not completely new. It's about 30 years in the making. So, over the past 30 years, science has been looking at how to, what's the genetic sequencing of viruses, looking for the proteins to target, writing the correct code for the mRNA, researching how to transport the mRNA into the cells, etc., without it being destroyed, ensuring that actually an immune response is delivered, and then also having that global IT infrastructure in place to allow that sharing of data. So if you look at the Oxford vaccine, the trials had thousands of people in Brazil, South Africa, England, and other places, and the only way they could actually run these trials was by having those infrastructures in place. So although not completely new, the technology is not new, the research has all been there, and this is now the first time that actually it's all come together for humans. There are some DNA vaccines and mRNA vaccines that have been approved for use in animals. So do these vaccines contain animal products? No, they don't. Neither the Oxford nor the Pfizer vaccine contain any animal products. Another question that is asked is, or a rumour that's spreading, are that they contain human fetus tissue or cells. Neither the Oxford vaccine nor the Pfizer vaccine contain any human cells. The Pfizer vaccine is completely synthetic in the production. The Oxford vaccine, when they're manufacturing it, they do use human fetus cells as part of the manufacturing process. Now, what's happening is they're using these cells from a human fetus that was aborted in the 1970s, not for vaccine reasons, but anyways, they're using these cells, which have over time they're literally just for scientific reasons, to produce the inactive virus that we talked about earlier on, that is going to then carry the DNA to then deliver it into the human cells. Once those viruses have been produced, the human cells are completely removed using a purification process from the actual injectable vaccine. Do they contain a microchip? Well, no. So there's 3 different types of rumours circulating. One is that they contain some kind of radio frequency identification device, an RFID, a bit like the contactless technology in phones to pay for contactless payments. No, it's not possible. Even the smallest type of chip it just would not fit through a syringe needle. Some people say that they contain a GPS tracking device as part of some kind of wider Bill Gates conspiracy. Again, no. All of these types of devices require some kind of power supply, so therefore, again, not impossible to inject via a syringe needle. And finally, some people saying that this is some kind of mark of the beast and will change people's faith. Again, there's no microchip. Religious people should bear in mind that, God does enable science and medicine. A reasonable question to ask is actually, could they alter your own DNA? Well, mRNA, it's impossible for it to alter your own DNA as it does not enter your cell nucleus. So the cell nucleus is where your DNA is, and that's the kind of brains of the cell itself. The DNA from the Oxford vaccine does enter your cell's nucleus, and that's where it then unravels and mRNA is produced. However, it is episomal, i.e. it remains completely separate to your own cell's genetic material and is not part of any integration, or doesn't become part of your DNA. And in fact, it is transient, so it's lost when the cells divide, with a range of kind of 7 to 42 days, thereby requiring those second doses for maximum immune response. It's not possible to get COVID-19 from this vaccine, from the vaccine itself. So they don't contain any of the coronavirus itself, only the genetic instructions for the spike protein. So therefore it's impossible to get it from the vaccine itself. That doesn't mean that you could not catch COVID-19 in the future, but you're not going to get it from having the actual vaccine. And the vaccines are considered halal by Muslim faith leaders. Really hope that you've found this a useful presentation. We have written this in good faith and tried to present the information as accurately as possible. Obviously, this is a changing situation, so things can change. It's important to keep up to date with credible sources of information such as the .gov.uk websites and the .nhs websites. If you've got any feedback or any further questions, feel free to get in touch with us on feedback@healthality.co.uk. You can also email us if you'd like a PDF copy of these slides.